right, good evening, everyone, and welcome to Eastern Connecticut State University. I'm Bill Salk, I'm the provost here, and I just want to welcome you all. We are incredibly excited about this conference, and we're really excited about the cannabis program here at Eastern. It was about 4 years ago that we decided we were going to start a cannabis program because we were looking, and Connecticut had not yet legalized it, but everybody else was moving in that direction. We knew it was a matter of time. Problem we had then was we didn't have anybody who could grow and teach people to grow cannabis. So I was telling Brian earlier, probably the greatest hire I've made in my time as provost is to steal him from another university up north and get him to come here. He has done a phenomenal job building the program we have and continuing to build the program as it grows into the policy side, into the business side, and things like that. So I want to thank Brian for really coming to Eastern and being kind of the driving force behind this conference and all that we do. We wouldn't be here without it. I also want to thank Amanda Irwin, so Brian has all the ideas. Amanda makes them all happen. You probably met her when you were checking in. This would not have happened without her, and I think tomorrow when it's over, just take all of next week off. Definitely. So again, welcome, thank you. I hope you have a great experience. The whole point of this is to bring together academics like our keynote speaker and Brian. Students, we love to see the students here. I'm glad that you made it for dinner, and I hope you have a great experience. And then really folks from the industry, because the way that this really should work as we move forward is to merge the academic side with the industry side, because there's a lot that we can do for each other. For the industry side, we can help you as you develop both your businesses and your plans. And for us, right, you can provide internships and valuable experience for our students. And so we hope that this is the first annual cannabis conference at Eastern, unless UCONN wants to host it at some point. We would certainly allow that to happen. But I hope it's also the beginning of a conversation among all the folks who will be here this weekend as we really see this industry grow and do good things within the state of Connecticut, particularly from the social justice side. So with that said, I will welcome you again, and turn it over to Brian to introduce Dr. Connolly to introduce our keynote speaker.
Thank you, Bill. I'm Dr. Brian Connolly, Assistant Professor of Biology. I'm the coordinator of the cannabis minor here. It's a real pleasure to introduce Dr. Gerald Berkowitz tonight. I've known Jerry for 20 plus years.
Something about that.
Through UCONN Plant Science, but also through the store's farmers market,
right,
where he comes to our farm stand and buys gold cherry tomatoes. And also, he's a very active member of the sustainable agriculture community in Northeast Connecticut. He also teaches a vegetable production class, which comes to our farm as well. So I've had many interactions with Jerry, Dr. Berkowitz. Yeah, it's really great to see his students out there. I think I almost killed one once with a hot pepper.
Right.
I had the Carolina Reaper pepper, which was once ranked the hottest pepper in the world, and there was a student who insisted on eating it in her class. But we got through that. Yeah. That was another experience. But Jerry has just been so supportive of this cannabis program. He's been generous and kind. He's been wanting to network with us and plug us into all the resources. We really appreciated his kindness and generosity. Just on a more academic, to talk about his background, Dr. Berkowitz has been a professor for about 40 years between Rutgers and University of Connecticut. He has gotten many awards for teaching and research. He's published so many papers, and he's just an all-around great guy.
Thank you, Brian.
Oh, you're welcome. But anyway, it's a privilege to hear his talk tonight. He's going to be talking about the molecular analysis of cannabinoid biosynthesis genes how hormones, environmental signals, and transcription factors can increase the THC or CBD content of cannabis, or you could summarize it as how to grow stronger weed.
Well put, Brian. I'd like to thank Brian and Eastern for being a leader in this area. And if you listen to the news, you're going to hear about lots of other colleges and universities following in Eastern's path, and Eastern's leading the way. And it shows. And I have a world of respect for what Brian's been doing here. So we all get to think about a very interesting plant tonight. In fact, the first review article I ever read about cannabis, the title was "The Plant of 1,000 Molecules." And we are just beginning to scratch the surface of what's going on in that plant. Because for decades, the industry has not published any of their genetics. Industry is putting out all sorts of things and doing all sorts of work, but they don't publish. And the federal government hasn't supported research to understand cannabis. They've supported research on how to eradicate it, but only now, just now, they're beginning to fund studies to understand how cannabis works. And I particularly am interested in how signaling works, how cells perceive signals. So that's my take of things. And I want to start out by mentioning the people in the lab who have done the work. Yi Ma has been with me. We've worked together for 10 years. She's was a postdoc. Now she's a research assistant professor. Peter Apicella, Peter graduated with a master's, got a job in the industry. Sam graduated and got a job in the industry. Gang did his PhD and is doing a postdoc in neurobiology. Lauren is working at an extraction facility, and Daniel has started a consulting business. There's a lot of opportunity for jobs in cannabis. And when I think of cannabis, I think that we're really talking about a plant that has a lot of unknown magical qualities, not to be, okay. But what I do want to say is that we really have a chance to look at the majesty of life, really how things work. And there's a lot that's not understood about cannabis. So I think that we're all here to study how life works. And I do want to pay an acknowledgement to the two police officers from Cheshire who were passed away in their funerals, who were killed in the line of duty. So as I celebrate life and we all study and learn about the magic of life, I just want to start by acknowledging that it's a shame that there's incidents like that. Well, my work is funded by a number of agencies and companies. CT Pharma is a medical company. I do suggest that Eastern get a hold of Adaptive. They sell LED lights, and they'd be really happy to donate lights to your greenhouses that are designed for cannabis. Fine Fettle Dispensary is, they have an operation in Willimantic, I believe, but they have a grow facility in Massachusetts. USDA has funded me to study this work, and I've had funding from NSF. Whoops, sorry. I want to advance this way. Okay. Well, first of all, we're going to study cannabis, but one of the beauty of research is we go to publications about how things work. So in my talk about cannabis, I'm going to use knowledge that's gained from orchid species, from snapdragon, tomato, sweet wormwood, tobacco, and our model plant, Arabidopsis. That's a plant that's sort of the white mouse for plant studies. So we do plenty of work with Arabidopsis, yeast, and I think we also are going to mention some animals, but we'll get to that later. Okay, so the first thing is why I asked Brian, he suggested I give this talk in a broad context. So I want to start with why we're here today. And why we're here has to do with Mitch McConnell, who's shown below in a field of cannabis. And it was Mitch McConnell when the Republicans, when he was head of the Senate, majority leader, in 2014, he inserted in the 2014 Farm Bill. Farm Bill comes up for legislation every 4 years. I circled in red. He inserted language that basically he was looking for a crop to replace tobacco in Kentucky. Tobacco was on the decline, and his state is agricultural. So he inserted language in the Farm Bill that said, I circled it in, I put it in a red square, that legalized the growing and cultivating of industrial hemp for research purposes in any state where it was legal. And just that was it was in the 2014 Farm Bill, the legislation to implement it in 2016. In 2017, because it said the word research, in Virginia there was 35,000 acres of hemp grown for research purposes. So in any case, my point is there's a lot of silliness with regards to the federal government and cannabis, and that's the beginning. So why are we here? And why has the federal government chosen to say that any cannabis that's below 0.3% THC is federally legal, and anything above 0.3% is federally legal? Has anyone in the audience heard of this or knows the basis for why the federal government has decided that? So I'm glad I included this. This is a paper from 1976. And in 1976, someone from the New York Botanical Gardens and a plant systemist from Canada went and surveyed the cannabis that was found in Canada, in greenhouses, whatever. And what they did is, this is 1976, and they found that the average total cannabinoids in the flowers and the leaves was 2%. 2% total cannabinoids, and they published this paper that said, we arbitrarily adopt a concentration of 0.3% THC to just separate what we found. And at the time, the total cannabinoids were 1/10 of what is going on today. So the federal government uses this paper as a basis for all the laws and issues regarding hemp and marijuana, which is, could you put it better, reefer madness. It's absolutely absurd to consider from a paper published in 1976 to regulate cannabis today based on that. And this is going to be the only talk probably ever presented that's going to include Mitch McConnell and Cheech and Chong in the same presentation. So I want to mention that back in the day, in 1976, they used to have what was called sinsemilla. And sinsemilla was unfertilized female flowers from a female cannabis plant. And back then, sinsemilla was the best cannabis you could find. And now today, my point in bringing up Cheech and Chong smoking their sinsemilla is that today, all our cannabis is from unfertilized female flowers. That's the standard. There is no leaves, or only leaves are part of the inflorescence. But my point is that from 1976 to today, it's a different world when it comes to cannabis. Okay. And by the way, people can interrupt me, and I'm not sure how much of this talk I'll get through, but we study the genes in the cannabinoid and terpene biosynthetic pathway. And our work focuses on identifying what's rate limiting the production of cannabinoids. We analyze genes and the regulatory motifs of genes that control their expression. We study how those genes are regulated, and we look at also the molecular basis for trichome formation, because it's the trichomes, you could see them here. Not all trichomes, by the way, about 20% of plants have these little hairs, trichomes, but there's different types of trichomes. In lots of plants, that lavender, tobacco, cannabis, that have glandular stalked trichomes. The ones down there, the stalk has a bulb. The bulb is actually an extracellular reservoir, and that's where all the action is happening, in the trichomes. And the proteins are made in cells just underneath that cap, but they're pumped, the enzymes are pumped into the bulb because THC and CBD are toxic. They're toxic to plant cells even. So they're made outside of the cell. They're made in the bulb. So that's point number one to learn. And I'm showing you a flower, and it's got some leaves attached to it. That's the inflorescence. And it's really, I show you a close-up of a sepal of a flower that shows the, let's see, which one is the pointer? Yes, so there you could see the trichomes on a sepal of a cannabis flower, and that's where all the trichomes are, and that's where all the action is. So first I want to mention that actually THC, CBD, CBG, all of these cannabinoids are generated as acidic moieties. They're actually THC acid, and these acidic groups with heat and time just come off non-enzymatically. So actually throughout the talk you're going to see references to THC or THCA. It's basically the same thing. In the plant it's made as THCA. It's effectively the same thing as THC. So what are the molecular switches? We're hunting for how the cannabis plant cells, the epidermis of the flower, what are the signals that cause these trichomes to be made? And then once the trichomes are made, what's causing the cannabinoids to be generated in those extracellular caps? So the first work that we did is we took hemp and we grew it in our greenhouse, and you put it, cannabis, most cannabis is day length sensitive. There are some varieties that don't respond to day length, but most cannabis, most commercial cannabis that's grown, what they do is they put it in a short day, like the fall, and when you switch the lights to short day, you initiate a flowering cycle. So that we initiate flowering cycles by changing the light period, and then we study each week, we study the flower as it develops. And this is another picture just showing you as the flower matures into an inflorescence that has trichomes. So the first thing we wanted to do was test our system. So we took flowers and we isolated plant material, and we checked for a cannabis gene. This cannabis gene I'm going to call GOT. We'll learn about it later. But we checked to make sure that as compared to the root, we see 100-fold increase in the presence of this gene in flowers. And we also see it somewhat in leaves. So we're just testing our system. There's a very complicated slide, and it shows you a complicated pathway because actually cannabinoids, the orange pathway, depend on fatty acid synthesis. There's an enzyme, fatty acid synthase, that generates different lengths of short-chain fatty acids. And it's the 6-carbon hexanoic acid that's a precursor. But also it requires other compounds, from the pyruvate, the cytoplasmic TCA cycle, requires all sorts of precursors. But the bottom line is that a compound in the cannabis pathway called olivotolic acid is combined with something called GPP. And this is the first interesting question that I scratch my head and think about. GPP, geranyl pyrophosphate, is a precursor for all monoterpenes. So in all plants that have terpene fragrances, all the monoterpenes are generated from this precursor GPP, and only in cannabis is this precursor to terpenes, geranyl pyrophosphate, also used for a different pathway, the cannabinoid pathway. So the geranyl pyrophosphate in these cells is got to be bifurcated, and nobody understands how the cells are going to do that. How are the cells going to know how much geranyl pyrophosphate should be diverted to monoterpenes and how much is going to be used to be making the first cannabinoid, CBG, cannabigerolic acid. So GPP, no one knows the answer to that question. Students could go to graduate school, study that, get hired by cannabis companies, make a lot of money. That's unknown. Something as simple as that. And I mentioned, I have a picture of the Phalaenopsis orchid here because I read a paper about this orchid and it had a very interesting point. This is a fragrant orchid, and this orchid smells wonderfully because it makes monoterpenes. And this orchid could be genetically engineered. So they took the enzyme that makes geranyl pyrophosphate and they increased its expression in this orchid, and it was more fragrant. So the implication here is that the monoterpene pathway and all of the monoterpenes, limonene, smells like lemon, all of these terpenes that give fragrances, they're all dependent on the geranyl pyrophosphate, and geranyl pyrophosphate rate limits the pathway. So it's rate limiting the terpene production, and it's also used in cannabinoid only in cannabis. It's an unknown question as to how it's regulated. So I'm going to make it simple and just get to the business end of the pathway where we have that geranyl pyrophosphate and it's condensed with olivotolic acid, OA, and it generates the first cannabinoid, CBG, and the cannabigerolic acid. CBG is now a pretty hot item. Lots of cannabis growers are growing strains of cannabis that just have CBG, so there's mutations in all the other enzymes, so it stops at CBG. But my point is that the enzyme that makes CBG is called geranyl pyrophosphate, olivotolic acid transferase. We call it GOT. So in any case, GOT generates CBG, and CBG is the precursor for all the main cannabinoids, CBD, THC, CBC. And this is a very interesting study. This is results of an experiment I'll get to in a second, but I want to mention Oregon CBD. Oregon CBD just gave, I think, $1 million to Oregon State University to study cannabis. Oregon CBD is a powerhouse. They're a major supplier of cannabis seed. And Oregon CBD, on their website, this is from their website, they say that all of their hemp, their CBD strains of cannabis, have their THC production turned off because they mutate a gene, THC synthase. And I took a hemp strain, cherry wine, a very popular hemp strain. Do you have cherry wine?
I do.
Okay, well, we can get you some. So cherry wine, very popular strain, and we measured the THC accumulation. This experiment right here is a felony. This is a hemp that's sold commercially, and in the greenhouse, the THC level rose to 1%. We cloned the THC synthase gene, and it had no mutation. So there's plenty of hemp out there that has THC, and what's more important is we're not 100% sure how the THC is generated in hemp. We're not sure. Larry Smart from Cornell, he's published a paper where he thinks that it's actually the CBD synthase that can make THC. Oregon CBD thought it was the THC synthase. And it could be, well, actually they think that it's the CBC synthase. At the bottom in red, they claim that it's the CBC synthase gene that makes an enzyme that makes THC in hemp. So it's not even known, but there's THC in hemp, in what should be CBD, no THC cannabis. And it's a real problem. This is a report from our Agriculture Economics, our Center for Food and Resource Policy, where they evaluated CBD hemp production in Connecticut, the costs and benefits. I just want to mention that CBD production is a $6 billion a year industry in the United States. In Connecticut alone, the first year that hemp was grown legally, there were 100 acres of hemp grown in Connecticut. And nationally, in this paper from Frontiers in Genetics, this paper estimates that about 20% of the legal hemp grown under Department of Agriculture programs has to be destroyed because it's too hot. It has too high. It has above 0.3% THC. So one important research question for you folks to go to graduate school and study is, how come hemp has THC? You have to legally test and then burn and destroy 20% of your crop. That's the average, and in Connecticut, certainly it's the case. I just did some math here. If you take the average value of a hemp crop is $30,000 to $50,000 per acre. The input costs are $20,000 an acre. If you conservatively estimated how much hemp has to be destroyed because it has unacceptably high THC levels in Connecticut alone, a conservative estimate is $200,000 a year. $200,000 a year in our little state of Connecticut. It's an important question. So now I want to go to a problem that I haven't been able to figure out yet, and that's what's the difference between hemp, between CBD synthase and THC synthase. So you can go to, whoops, oh no, I want to minimize this. I want to call up this. So this is from the Swiss Protein Database, and let's see. So this is THC synthase, and it has a cofactor FAD, but this is THC synthase. And you know you can take we took CBD synthase and threaded it through the crystal structure of THC synthase, and we could not determine which amino acids in the active site cause CBD and THC. We need better computational biology than what I was able to use to try to look at this structure. And I generated a structure of CBD synthase, but we couldn't tell the difference. We couldn't identify which amino acids were important that were critically different in THC versus CBD synthase. Now I'm introducing you to the trichome, the glandular stalked trichome, which is present in a lot of plants. And the glandular stalked trichome has a series in cannabis of cells called a rosette of gland cells right underneath the trichome head. You see on the left, on my left, the extracellular reservoir, and it's the cells just underneath there, these rosette of gland cells that have all the genes that are making the enzymes that are doing all this cannabinoid biochemistry. So we purify these rosette of gland cells. This shows you a sample of our preparations, and we study the genes in those gland cells. So this is a complicated analysis where we took all the measurements of THC, CBD in marijuana. We did this at CT Pharma Medical Marijuana Dispensary. So we worked with them, and we took their marijuana, and we took weekly measurements of all of the precursors, all of the enzymes, and we found something very interesting. What I want to point out to you is that this shows the correlations over the 7-week flowering period between the THCs and the enzymes that make them. And I want to point out to you that there's a highly negative correlation between the enzyme that makes THC synthase and THC levels in the plant. THC synthase, the enzyme that makes it, is nothing to do with rate limiting the production of THC. That's the first thing that raised my eyebrows. So there's a highly negative correlation. We'll see why in a minute. But I do want to point out that in this analysis, let's see, GPPS is highly correlated. That enzyme that makes geranyl pyrophosphate, the precursor to CBG, is rate limiting the pathway. That's what this data suggests. The previous slide is a giant analysis of many experiments like I'm showing you now. So this is the weekly and levels of THC on the left. This is the weekly levels of THC, and you could see as you flower towards week 4 after putting the plants in a flowering cycle light, you see the THC rising to like almost 25%. That's marijuana. That's what modern marijuana has. And look at the expression level of the enzyme that makes it. It's doing nothing. It's not changing at all. That's why there's no correlation. But if you look over to the analysis of the enzyme that makes CBG, this is the enzyme, that GOT enzyme. It increases 30-fold during flowering, and its peak is just in rise with the rise in CBG. And as THC is generated towards the end of the flowering period in a high extent, you start seeing CBG drop. And if you look at the level of CBG, it's 1/10 of the level of THC. So my point is that the enzyme that makes THC is not important. Plant breeders, cannabis companies, should look at how this enzyme GOT makes CBG, and we should look at its promoter and how it's regulated. That's the first thing we learn. This is just an example of the many, many other genes involved, how they all are upregulated during the flowering cycle. So not only is CBG level 1/10 of THC, and this is all new information, it's not been published, and I think it's going to be interesting for the cannabis industry. So not only is the level of CBG 1/10 the level of the end products, there's an interesting paradigm that CBG is made in the plastid envelope, where olivotolic acid and geranyl pyrophosphate are combined at the plastid envelope. But THC is made outside of the cells in the bulb of the trichome. So not only is CBG rate limiting, but nobody knows how the heck it goes from the chloroplast out to the reservoir. What are the transporters? How does it move? Nobody knows. Nobody's studied any of this. Nobody's published in refereed journals any of this. So we looked at a family of transporters. These are called ABC transporters. They use ATP, an energy compound. So they're called ATP binding cassette transporter. So we were thinking, and they generally transport anything. So we were thinking this is we were thinking that this is a good possibility. So we went and looked, and there are data sets of all the genes in the trichome gland cells. So we went and looked at the online databases, and we see that some of these ATP, these transporters are highly upregulated as the flower matures. So maybe they're candidates. But just, 2 weeks ago, a brilliant paper was published by Livingston, and one of my graduate students just goes gaga over Livingston. And that's even before this paper was published. So this paper was published just 2 weeks ago, and it informs us a whole lot. This is a compilation of, I think, 110 different electron micrographs, where they put it all together to try to study. These are those gland cells right underneath that secretory reservoir. And what they did is they saw how there's plastids. Remember, that's where CBG is made. They saw how the plastids are accumulating right near where that extracellular reservoir is. And here's a close-up of one of those plastids, and it shows you how GPP is made into CBG, and they have an unknown, they mention ABC transporters here, but it's completely not known how CBG gets out to that extracellular bulb where THC is waiting to make it. So all of that is not known, and it's absolutely critical for increasing cannabinoid value. Let's put cannabis value. So this is a very interesting picture of what's called trichome morphogenesis. There's not a whole lot of research done. There's some research done with tomato and sweet wormwood and Arabidopsis, but we're just now beginning to understand all the signaling that causes an epidermal cell to morph, to undergo morphogenesis, to initiate a trichome. And when you look at the slide, think about this. How come when you cut your finger, a liver doesn't grow? What's the answer to that question? How come? I don't know the answer to that question, but I imagine that there must be signals from skin cells that are telling the cells that are growing, the stem cells, to turn into finger cells. So there's messages that signal, because your skin cells that are growing, they have the entire chromosome, all the genes in them. How come only skin cells are turned on? Think about that. How come a liver doesn't grow on the tip of your finger? That's what this is about. So what are the signals in the epidermis that tell a cell, okay, initiate the pattern to grow into a trichome? What are those signals? And knowing those signals might be very valuable. That's one of the things we study. So let's see. So this is trichome morphogenesis. An epidermal cell gets signals from other cells, and those signals tell it become a trichome. And there must be signals from the trichome, from this cell, to tell other cells not to become trichomes. All of this is waiting for you folks to go to graduate school and study. So I'm just saying, there's so much not known. It's amazing to me, because the federal government hasn't supported research companies don't publish, so all of this is unknown. So we went to Snapdragon. A student of mine found a paper in Snapdragon where they took a gene from Snapdragon and they put it in tobacco. Tobacco is an easily transformed plant. It's very hard to genetically engineer cannabis. It can be done. Companies do it, but they don't publish, and the current publications are garbage. They're really not the stuff that is published. So cannabis is not a plant that's easily transformed. Plants differ in their ability to be engineered. Some plants are easy, like tobacco. Some plants are not, like cannabis. So in this paper, they took a gene from Snapdragon, from the petals of the flowers, and they put it with a promoter that causes it to be expressed in all cells, and they put it in tobacco. And here's what they found. This is a tobacco untransformed. Here's a transgenic tobacco. This is a trichome growing in tobacco that doesn't have any other messages in it. And so this is a trichome. And here, when they put the gene from Snapdragon in tobacco, this is what they found. A forest of trichomes, a literal forest, all different types. These are non-glandular, these are glandular stalk trichomes. So we went to Snapdragon and said, let's go find a gene that's similar to the Snapdragon gene in cannabis. And this is our cannabis sativa gene that's similar to the Snapdragon gene. We used PCR and we identified that it's highly expressed in flowers, and it's even expressed in the trichome. We don't know what it's doing in the trichome because it's in the epidermis as a signal to make a trichome initiation. We don't know what it's doing in the trichome yet. This is another one of those matrices where it includes that gene mixta. That's the cannabis gene that is similar to the gene that makes trichomes in tobacco from Snapdragon. So we look at the expression of mixta in cannabis, and it's highly correlated, as opposed to THC synthase. It's highly correlated with the enzyme that makes geranyl pyrophosphate. So there's all these positive correlations. My point being is that the generation of trichomes in cannabis is where the money is. How do you get trichomes to be increased? Mixed is one possibility. So here we took the gene from mixta, and we took the promoter from the gene. We took the full-length gene and we fused it to green fluorescent protein, and we expressed it in, I think, Arabidopsis. And you could see that it's in the nucleus. This green light means it's in the nucleus. So this cannabis gene can get into the nucleus. Evidence number one. Evidence number two, we used yeast. Yeast is a wonderful tool to use in biology, because yeast, you can get hundreds, thousands of mutants. You can simply order and get for free from the yeast repository. I think it's at Yale. But you can order any yeast mutant you want. So we ordered a yeast mutant that is lacking a gene that binds to DNA. Let's see, what is it? So the yeast mutant can't grow unless you supply it with a transcription factor. Transcription factor is a protein that signals what to turn on and off. That's how come your cells don't grow liver, because they don't have a transcription factor that signals become a liver cell. They have transcription factors that say become a skin cell. So we took that gene from cannabis and we put it in yeast, and we put it in yeast that has a mutation that it requires a transcription factor in order to grow when you have selected media. If you have just tryptophan lacking in the media, the yeast will grow with or without anything. But this yeast mutant, if you lack histidine and tryptophan, it requires a transcription factor. So we use the yeast transcription factor, and you could see it grows. And then we use the cannabis mixta, and we see it grows. So these data demonstrate that this cannabis mixta gene is a transcription factor. And by the way, UCONN patented it, and it's available for this, this gene is available as a tool to increase trichomes. So let's look at what it does to trichomes. This is a tobacco leaf where we express the cannabis gene in the tobacco. And I got to tell you, to me, this guy looks like an old man. You can see it's hairy. These are trichomes. That cannabis gene, when we put it in tobacco and use a promoter that will express it all over, the tobacco becomes hairy. These are trichomes. So this is all evidence put together that that cannabis gene is a trichome initiation gene. So now, my graduate student held up a tobacco leaf expressing this cannabis gene and took pictures. And this is what the picture, we're not really good photographers, but but take a look at these trichomes. They're like monsters. They have branches, they have bulbs, and remember that mixta is expressed in the trichome. So we don't know anything about what's going on here, but we certainly know that we found a cannabis gene that causes trichomes to be developed. And here you see we tagged it with GFP, and it's in a trichome. So you could see it lighting up in the trichome. So now we take electron micrographs of tobacco. So this is electron micrographs. This is just normal tobacco, and this is the trichomes in a normal tobacco leaf. Oh my goodness, I'm running out of time. But what I want to mention is here is an epidermal cell undergoing trichome morphogenesis. It's getting the signals to make a trichome. And when we express that cannabis gene in the tobacco, we make far more trichomes, and we make these monster trichomes that are branched and giant and have all sorts of trichome heads. This is looking at the promoter of the transcription factors. There's mixta, and you see the promoter is the region upstream from the coding sequence that signals when the gene should be turned on and off. So transcription factors bind to the upstream, the promoter of genes, and that's what causes the gene to signal, okay, turn it on. So we looked at the promoters of all the cannabis biosynthetic pathway genes. We cloned some of these transcription factors, and this mixed transcription factor has a gibberellic acid binding domain. The promoter of mixta has a gibberellic acid binding domain. So we went to greenhouse cannabis plants and we sprayed it with gibberellic acid. Lo and behold, this is the normal upregulation of mixta, a 30-fold, 40-fold increase in week 4, right before the trichomes start developing. And when we sprayed gibberellic acid, it prevented that. So in this case, the binding element prevents expression of the gene. This hormone, gibberellic acid, it binds to the promoter and prevents RNA polymerase from making the message. This is a different plant. This is sweet wormwood. Sweet wormwood is a focus of a lot of study because it makes artemisinin. Artemisinin made in trichomes is an anti-malarial drug. So sweet wormwood is a very important plant. And this is a wonderful paper that a different graduate student read and brought to me. And this paper reports how, in this case, they had a gene called HD8. And in sweet wormwood, what happens is when they knock out HD8 by using what's called RNAi, you can put an RNA into a plant that will prevent expression of the gene. So they knocked it down, and and they showed there's less trichomes, and then they transformed it with this gene called HD8 in sweet wormwood that causes increased trichomes. So we went and looked for the cannabis homologue of HD8. We call it HD1 in cannabis, and it's a transcription factor, a certain type, called a leucine zipper transcription factor, and it's highly expressed in flowers as compared to the root, and some in the leaves. So we have another transcription factor, and here is the THC content in marijuana as the transcription factor is being generated that makes trichomes. And it's these trichomes and those balls at the top, that's where all the action is. This is a scanning electron micrograph of tobacco expressing that second transcription factor. And you could see, this is transgenic, you could see that it's got all of these, again, monster trichomes, and far more than the wild type. So in the paper on sweet wormwood, they sprayed a plant hormone called jasmonic acid. They sprayed jasmonic acid, a plant hormone, on wormwood. And they found a big increase in expression of trichomes. So we took methyl jasmonate and sprayed it on cannabis, and we got an increase in, we did this at CT Pharma, we got a 20% increase in THC just from applying a plant hormone, which is innocuous. It's not a pesticide, not an herbicide, it's just a signal. So this is a representation of a gene where you have the coding sequence that codes for the message that becomes a protein, and this is the promoter region. And this is where RNA polymerase that makes the message binds. And it's not so simple. The promoters can be pretty complicated. This is another representation of a promoter of a gene, and you could see how all sorts of factors bind to it to regulate expression. So so this is, again, a picture of epidermal cell undergoing trichome morphogenesis to become a glandular stalk trichome. And we studied that gene we think is rate-limiting THC production, that geranyl pyrophosphate olivotolic acid transferase got. And we looked at its promoter, and we looked at all the hormone-responsive elements in its promoter, and it's got a gibberellic acid, abscisic acid, salicylic acid, ethylene, cytokinin. The one thing it doesn't have is a ABA response. No, it has an ABA. What is it? It doesn't have an auxin responsive element. So we did experiments where we saw there was just one element that bound the hormone salicylic acid. That's another plant hormone. So we took plants and we sprayed salicylic acid on the plants, and this is the relative expression of that got, and we increased 100-fold the expression of these cannabis genes from using that salicylic acid. And now we come to interesting experiments where we use animals. We use a gene from firefly, and we use a gene from sea pansy. I think that's what it is. They both light up. So they have genes that luminesce. So we took the promoter from that cannabis gene, that gene that we think rate limits THC production. And we took just the promoter and we constructed constructs that had the firefly gene that generates light, and then also the sea spray, the sea pansy gene. These are two different genes, so we could look at the relative light generated by the luciferase controlled by the firefly versus the sea pansy genes driven by that cannabis promoter. And what we found was that when we spray Salis here, we took those genes and we put them in Arabidopsis protoplast. And we studied giving salicylic acid to the protoplast and studied the light. So this is the light generated by the promoter that is from what we think is the most important gene in the pathway. And it shows that salicylic acid can increase its expression many-fold when the gene is expressed in Arabidopsis. So this is a totally different system. But what was nice is there was only one salicylic acid responsive element in the promoter of what we think is this very important gene, so we could easily mutate it. So we knocked that out, and salicylic acid no longer causes a change in the expression of the cannabinoid gene. So what we're revealing here is that we think that we're on to how the most important gene in the cannabis pathway is regulated and how the promoter is responding to plant hormones. Now, my last group of slides has to do with the following. Plants are engineered by evolution to respond to the ratio of red and far red light. That's a way for them, then they can do shade avoidance. When plants see a change in the ratio of red and far red light, it's a signal for them to grow and grow away so they can get light. So plants respond to red, far red light. And here's an example where with no far red light versus far red light. So plants are responsive to the ratio of red to far red light. So we got donated to us from Adaptive. They sell cannabis LED lights. And we got the latest generation. We got $60,000 of lights donated, where you can program these lights to have any wavelength you want. You can knock it out, you can increase it. And these lights are great to study cannabis. So what I want to show you is that plants have pigments like chlorophyll and phytochromes and other pigments that all absorb light. And I want to point out that phytochrome absorbs in the red and the far red wavelengths. So we took these cannabis lights and we changed the ratio of red to far red light because we know plants are responsive to that. And actually my graduate student had heard that if you grow plants with cannabis with LEDs versus mercury halide, someone published a paper showing that there are much different effects. But they didn't prove that it had to do with this because it was different lights with all different spectrum. So what we did is we took these cannabis grow lights and we changed the far red light. This is the regular cannabis, and now we changed it to knock out the far red, to reduce the far red light. So we changed the ratio, and lo and behold, we have more cannabinoids. So the plant is responsive to the ratio of red to far red light, and the flowers are even bigger. So all of this is just the beginning. And you guys, it's a wild party of biology. There's so much open to study. And thank you so much for your patience. And I couldn't be more excited that Eastern is the focal point of some great biology, because this all work needs to be done. And students need to hear about this and think about it, and the government needs to help us uncover and discover how life works even more intimately. And thank you so much for your time, your patience. Any questions? It's a long talk. Yes?
I've got one. I'm coming from an insect side,
Mm-hmm.
right? And a lot of the chemicals you mentioned obviously would be And I also noticed you had jasmonic acid as one of your...
Hormones.
Correct. So that is elicited when there is damage.
Yes.
So my question is, you've done the experiment with the plants to see if you happen to damage the plant outside of where the fluorescence is, and ask the question because it's so volatile.
So that's a great question, and we haven't, and it's not known. But it's probably, jasmonic acid is known, as you say, to be a stress response hormone. So there's no question that there's likely that insect damage is probably caused. And actually, there isn't a lot of published evidence as to what the heck trichomes are doing. Not just in cannabis, but in general biology. 20% of plants have trichomes. And many of those species, including cannabis, have glandular stalk trichomes where they have a reservoir. And that reservoir, again, the genes are in the cells. The enzymes are made in the cells, but then the enzymes are pumped out into that trichome head. But in any case, there's no question that you're onto something. Jasmonic acid and insect issues is absolutely a likely scenario. And I don't know, nobody really knows, did the cannabis plant evolve trichomes to have cannabinoids for us? Has anyone ever, what's the name of that great book, Omnivore's Dilemma? Anyone ever read Omnivore's Dilemma? They really talk about how, like, tulips are telling humans what to do. And I don't know, but why does cannabis have trichomes? Why are they making cannabinoids? There isn't a really strong answer in the literature as to what they're doing. They're toxic to the plant cells. They have to be made outside in those bulbs. So I think you're onto something. Anyone else? Yes, BJ.
Your talk made me feel very high.
Yeah. Well, it's exciting. There's so much to discover. It's remarkable.
But my question is, I think many of these questions can be answered by transgenic technology. Did anyone try different strains of cannabis that can be transgenic?
Yeah, there's maybe 3 or 4 publications. Your student is now working in Yi Lee's lab. Yi is an excellent transformation technologist. And I showed him the 3 or 4 papers of cannabis transformation, and he said they're all garbage. So what I'm letting you know is I think industry knows, but the referee publications are really lacking in terms of engineering cannabis. To be able to study it, because as you imply, we really need to. When I went to my patent office about patenting these genes, they went, well, you proved it in tobacco, but you need to really prove it in cannabis to make the patent valuable. And that awaits more study and better technologists than I. So good question, VJ. We really need to improve our ability to engineer cannabis so we can study it. It's not as if we need to engineer it necessarily to generate transgenic cannabis. Well, thank you all for your patience. It's a long evening. It's exciting to think about the scientists you'll hear from tomorrow. We're going to hear from Brian and some other folks from UCONN. So thank you all, and I wish you all a pleasant evening.